This is part 3 of 3 looking at chapter 8 on photosynthesis. So in the first part of this chapter we kind of looked at the general overview of photosynthesis and how it fits in with the ecology and the biosphere. In part 2 we focused on the light reaction, which is the first reaction within this photosynthetic process. So just to review quickly, the light reaction happens in the cycloid membrane, so that's the location. The inputs are light, so photons of light coming from the sun, specifically visible light, so the colored light. We also needed to take in water to get electrons and hydrogens. That water was split up by photosystem 2 in order to get the electrons and hydrogens, and then oxygen is released as a waste product. So these inputs are used, and they're used to create ATP and NADPH. So basically we're taking energy and light and converting it into the energy of ATP and NADPH. So this section we're going to focus on the second part of photosynthesis called the Kelvin cycle. This is where we're going to bring in our carbon dioxide. It's coming in at the top of the cycle. We're also going to use all of our ATP and NADPH that we created during the light reaction, and we're going to use the energy and hydrogens and carbon dioxide in order to make a carbohydrate or a sugar. The Kelvin cycle, or the Kelvin-Benson cycle, depending on the book you're looking at, it's named after the people that figured this cycle out. So what we're going to do is we're going to take our ATP and our NADPH, and we're going to use it to make an organic molecule or carbohydrates. So our carbon dioxide is going to be incorporated with hydrogens from our NADPH to create these carbohydrates. And this molecule that we're going to be creating, this is a precursor to all other organic molecules, so then the plant can use this organic molecule created during the Kelvin cycle in order to produce amino acids and other carbohydrates and nucleic acids and lipids. This carbohydrate that's produced, it can be converted into starch, which is how plants store energy that they then can use later for reproduction and growth. So this is really where we're starting to get these nutrients that are being created, or these carbohydrates that are going to fuel all living things. This process requires a massive input of energy that's going to be created during that light reaction. For every 6 carbon dioxide molecules that are turned into an organic molecule, we need 18 ATPs and 12 NADPHs. And again, notice that we need a few more ATPs compared to NADPHs, and we talked about that issue in the previous part. So if you don't remember that, make sure to go back and make sure you understand how those extra 6 ATPs are actually created during the light reaction. This carbon dioxide, we're going to use all this energy and all these hydrogens off of our NADPH, and we're going to produce a molecule called glyceraldehyde 3-phosphate, or G3P. This is that molecule that's going to be used to create basically anything the plant needs in order to reproduce or grow or to do any other thing that it needs to do. So glucose is not directly made using the Kelvin cycle, however, usually we refer to glucose being created for this. So like I mentioned, this G3P molecule that we're going to be creating, it can be used to pretty much build anything. So we can use it to create amino acids, we can use it to make glycerol and the fatty acids in order to make lipids. The G3P can be converted into glucose, which can then be used to build up larger macromolecules like cellulose, sucrose, or starch. So this G3P is really, really important to create all of these different biological macromolecules. So the Kelvin cycle is broken down into 3 phases. So the first phase is called carbon fixation. This is where we're going to take our carbon dioxide that's brought into the plant cells, and we're going to incorporate it into a molecule called RUBP. And the enzyme that does this is called Rubisco. So the RUBP is a 5-carbon molecule. Carbon dioxide is a 1-carbon molecule. They get hooked together with this Rubisco enzyme. We end up with a 6-carbon intermediate molecule that is then split into 2 3-carbon molecules called 3PG. Those 2 3-carbon molecules are then going to be reduced in order to create our organic molecule. So we have a reduction process and the carbohydrate production. So we're going to use our ATP. We're going to convert our 3PG molecule made during carbon fixation into a 1,3 bisphosphate glycerate molecule. The NADPH, we're going to take hydrogens off of the NADPH and use it to produce our G3P. Remember, G3P is that molecule that we're really interested in. That's the precursor molecule to every other biological macromolecule. So 6 carbon dioxide molecules are brought in during carbon fixation. We use up 18 ATP, 12 NADPHs, and we end up with 12 G3Ps, 12 of these precursor molecules that we want. 2 of these G3Ps exit out of the Calvin cycle, and they're used to create carbohydrates, amino acids, lipids. The other 10 out of the 12 are going to be used to regenerate our RUBP that we used at the very beginning of this process. So this regeneration process, this is our 3rd phase, regeneration of our starting RUBP. So those 10 G3P molecules are converted into 6 RUBPs using 6 ATPs. So again, remember this is a cycle, so we have things that are coming into the cycle, things that are exiting out, and we always end up regenerating our starting molecule. In this case, that'll be the RUBP. So here is that process in a diagram, and I have the 3 phases labeled. So if you look up where it says phase 1 up there, so we're going to have our carbon dioxides coming in as an input at the very top. So carbon dioxide has 1 carbon. We're going to fix that carbon dioxide to our RUBP using the enzyme Rubisco. And then we're going to have 5 carbons from the RUBP plus another carbon from the carbon dioxide to create a 6-carbon molecule that's split in half, and we end up with 2 3-carbon molecules called 3PG. Okay, so remember that's phase 1, that's the carbon fixation. Phase number 2, we're going to use energy, so we're using ATPs, we're going to use NADPHs in order to reduce our molecule. So remember, reducing means to add on hydrogens to our molecule, and that's going to create an organic molecule. So remember, organic molecules are when you have carbons and hydrogens attached together. So phase number 2, we have the reduction of our molecule and generation of our carbohydrate. So we're going to end up with 12 G3P molecules at the end of phase 2. 2 of those exit out, and they're used to make glucose and other sugars and other material for the plant. The other 10 out of the 12 are going to go into phase 3, the regeneration of our RUBP. So here's where we're going to use up a few more ATPs in order to regenerate our RUBP. Okay, so again, we have our Calvin cycle, things are entering in, they're exiting out, and then you get back to your starting point. Alright, so that was the process of photosynthesis. So again, this image is probably the most important image out of this chapter because it really summarizes what we've been going over so far. So this Calvin cycle, the Calvin cycle that we've looked at, this is called C3 photosynthesis. And about 90% of plants do the C3 photosynthesis that we looked at. However, we're going to look at a few, two different variations that can occur, and it really depends on what type of environment the plant is adapted to for what type of photosynthesis it's going to perform. So certain environmental conditions can influence the efficiency and the way that the Calvin cycle works. The light reaction pretty much is the same in every type of plant species, but the Calvin cycle has little variations to it. So different things that can affect how the Calvin cycle works. We have the light intensity, temperature, and then also water availability. So like I just mentioned, we focused on a type of plant called C3 plants. And again, about 90% of plants are C3 plants. And in C3 plants, that enzyme rubisco, that remember is the enzyme that is used during carbon fixation that hooks the RUBP and our carbon dioxide molecule together. So generally in just kind of normal environments, the rubisco, that enzyme functions as a carboxylase. So it works just like how we've been talking about. So the RUBP, which is 5 carbon, gets attached to carbon dioxide, which is another carbon. So we have a 6 carbon molecule that gets split in half, and we create 2 3 carbon molecules. So these are called C3 plants because they make this 3 carbon molecule during that carbon fixation process. And again, most plants, about 90% of plants use this C3 Calvin cycle. So plants that are in moderate temperature, moderate rainfall, moderate light intensity. However, in some environments, rubisco can also act as an oxygenase, the oxygenase. So instead of RUBP being attached to carbon dioxide, we have an oxygen that's attached to your RUBP. And this eventually leads to that carbon dioxide being released. This is called photorespiration. This is a very bad thing. We don't want this to happen. We don't want photorespiration to happen because we want carbon dioxide to be fixed into the Calvin cycle. We don't want it to be released back out into the environment. So what's occurring is that we're using oxygen, which isn't even what we want to be using, and it's liberating carbon dioxide. So it's a very, very wasteful process. This photorespiration, it's more likely to occur when you have really hot and dry environments. So more of the extreme temperatures or really low rainfall. And it's also favored when carbon dioxide is low in the atmosphere and oxygen is really high. So there are certain environmental conditions where this photorespiration will take place. Remember, this is something that we want to avoid. We want to avoid having oxygen attached to our RUBP because it's wasteful. We're not going to get any carbohydrates being produced. So one way that plants can kind of deal with this photorespiration process is by using something called the C4 process. So here we have C4 plants. So C4 plants, instead of fixing carbon to create a 3 carbon compound, they fix carbon into a 4 carbon compound. So they make a compound called oxaloacetate. So this is the first step in the carbon fixation. And besides just making this 4 carbon compound, they do this in 2 different types of cells. So these leaves in the C4 plants, they have 2 cell layer organization. So they have a type of cell called a mesophyll cell. This is where the CO2 enters in to the leaf through the stomata, those little openings on the bottom of the leaf. That CO2 is fixed into a 4 carbon compound, and this process does not promote that bad photorespiration. So you want to fix the carbon dioxide into a 4 carbon compound. That 4 carbon compound then moves into another type of cell called the bundle sheath cell. So our 4 carbon compounds transferred in to the bundle sheath cell. It releases that carbon dioxide molecule and allows it to attach onto the RUBP. So here we have the C4 process shown in this diagram. So we have our 2 different types of cells. If you look on the diagram on the right hand side, So the green cell, that's the mesophyll cell. So this is where our carbon dioxide is going to enter in. It's going to be attached to a PEP molecule to create our oxaloacetate, our 4-carbon molecule. Once our carbon dioxide is attached to oxaloacetate, it gets converted into malate. That enters into our second type of cell called the bundle sheath cell. So once that 4-carbon molecule enters into our bundle sheath, it releases the carbon dioxide and allows it to enter into the Calvin cycle and get attached to the RUBP. So we're just separating our carbon dioxide into different types of cells, and that's going to keep the oxygen out of our bundle sheath cell, so that we're not going to have that photorespiration process, that thing that we don't want to happen. Another variation besides the C4 plants is we can have CAM plants. This is Crasslucian acid metabolism. So they basically do what we just saw. They create a 4-carbon molecule, and instead of having different types of cells, they do this at different times of the day. So what happens is that CAM plants, they're going to open their stomata at night when it's cooler and there's more moisture in the air. This allows carbon dioxide to enter in to the leaves. It gets converted into the oxaloacetate, into the malate, so that we have our 4-carbon molecule that's created. So it does this at night. And then during the day, when it gets hot and dry, the stomata are going to close, so they conserve water. That oxaloacetate is converted into the malate. The malate is broken down, it releases that carbon dioxide, and then the carbon dioxide can enter into the Calvin cycle by attaching to the RUBP. So our C4 plants, they separate it in 2 different types of cells, so they separate it by space. CAM plants, they separate it based on time of day, or they do a temporal separation of capturing our CO2 and then bringing it into the Calvin cycle. So here we have our C4 plant on the left-hand side, and we looked at that image. A really good example of a C4 plant is corn, which is a really big agricultural crop. So corn is a C4 plant. It's really good at surviving drought and really hot environments because it can do this special extra little step before the Calvin cycle. In more extreme environments, like a desert for example, this is when we have our cactus. They're a CAM plant. So they're going to bring in carbon dioxide during the night. When it's cooler, they'll fix it into the oxaloacetate. And then during the day, that carbon dioxide is released and brought into the Calvin cycle. So in summary, we looked at the process of photosynthesis, and then we also looked at 2 little variations that can occur in the Calvin cycle, those C4 and CAM plants. Remember, 90% of plants, they do the C3 process that we looked at in detail. We do have those little variations where plants have adapted to really dry, hot environments to be more efficient and to cut down on that photorespiration problem. So again, just a reminder to really make sure you know this. This is basically the summary for this chapter. It goes through the photosynthesis process where all of our reactants come in, where our products come out, where the different parts of the pathway take place within our plant cells in the chloroplast.